Next we're covering very special cellular organelle. It's called ribosomes. So this is uniformal. It's present in all living cells, ribosomes. No cells without ribosomes present. It's very important. It have important function to produce different proteins. So these organelles, why it's very special, it's referred as non-membranous organelle. Non-membranous, it means this organelle ribosome not covered with membrane, but it's even present in the eukaryotic cells. So this is why this organelle very special. It's in the prokaryotes and bacteria, in the plants and animals, everyone have it, ribosomes, and it's non-membranous. So let's see what is the structure of this. So it's made up of the two units, large unit, and here showing the red, and small unit. It's in here in the diagram, it's showing the blue. And these units of the ribosomes, they made up of the different ribosomal RNAs, what not show kind of in here. But function always the same, to produce protein. And we will talk in the details later how they producing, how they functioning, yes? But these subunits, large subunits and small subunits, when they attach to each other, like on this diagram, they together, they call polyribosomes, when both large and small subunits attach together. And when this happening, it means that these subunits right now reading this messenger RNA. So this molecule in here between kind of them, this is messenger RNA. And when these subunits moving, they reading kind of, they moving through this messenger RNA, then from the cytoplasm coming and attaching to these subunits and to the messenger RNA, the other type of the RNA, it's called T, transport RNA. And in here it's green. And this transport RNA, each transport RNA holds different type of the amino acids. In here, for example, this amino acid. And when this attached and reading this information in here, nucleotides, so in the messenger RNA molecule, they attaching the amino acids to the other amino acids, what already was attached to each other. And different amino acids attached to each other, it will be what? This will be our first structure of the protein. This is the protein, how they form. The proteins made up of the amino acids. So overall, without little details, so ribosomes made up of the two subunits, large and small, and the main function is to produce proteins, synthesize different proteins. And these ribosomes, they could be free, how they call free ribosomes. In this case, they just freely located in the cytoplasm. And on the other hand, ribosomes could be membrane bound. It means they attach to the membrane of the rough endoplasmic reticulum. And this is why function of the rough endoplasmic reticulum is to synthesize proteins, because on the surface they have ribosomes. And in here again show some of this functioning, how two subunits of the ribosomes, they movements of the ribosomes, so these two subunits, large and small, they moving this way. And this is the molecule of the mRNA. And in here we see different nucleotides, guanine, uranyl, cytosine. And this is a blue, dark blue structure. This is transport RNA. Each transport RNA has special site to recognize different type of the nucleotide in here. For example, 3 cytosines, and each of them holding one amino acids, different type. So different this transport RNA, they will come, they will attach in the complementary way to the nucleotide, and based on this recognition of the nucleotide, different amino acids from the transport RNA in here, what is bringing this, will be attached together in this long chain. And this is long chain from amino acid is a protein chain. So overall, how production of the proteins happening? So in here we have part of the nucleus. So this is the nucleus. Below in here light yellow, this is the cytoplasm. So in this inside of the nucleus, we're looking right now inside of the nucleus. We have DNA molecule. In here show blue, double helix DNA. And during protein synthesis, this is double strings of the DNA will be open in the fork like this. 1, 2 strings, they open. And based on these different nucleotides, what in here will be produced, produce mRNA. So this is just primary transcript, they call it here, red one. And then after processing, it will be in here, long red. This is our messenger RNA. It's called messenger because it's carry message from nucleus. This message through the nuclear pores will leave the nucleus inside of the cytoplasm. And this is where is our messenger RNA in the cytoplasm in here. This is our messenger RNA. It's bring message in the cytoplasm that we need to produce protein which will have this specialized amino acids, but it will be based on different, this is nucleotide sequence. So this mRNA have different sequence, different nucleotide in here. And then ribosomes, which show in here large unit, small unit. This is gray structure, large unit and small unit of the ribosomes. They will attach and they will move. Ribosomes is moving through this is messenger RNA and different in here transport RNAs which holding different amino acids. They will come, they will recognize this is nucleotides on the messenger RNA, and they will attach these different amino acids to this chain of the amino acids, which later on will be protein. Yes, when they more and more amino acids attaching, this is what will be protein later on. So this is how productions happening of the protein. From DNA, information will be created in form of the messenger RNA, then it will leave, and ribosomes will read this is information in form of the sequence of the nucleotide and the mRNA. Ribosomes can read this information, and based on this, they will produce, help to synthesize protein. Here, chain of these amino acids. So again, this is just showing the kind of more higher magnification, how this synthesis of the peptide or proteins was made. This is the ribosome, shows this is color. This is a messenger RNA in here. This is different nucleotide. This is look like teeth. Different type of the nucleotide. And this is our tRNA, transport RNA, which also have this specialized teeth-like shape, and it's also nucleotide, and each attached to different amino acid. So this will be reading. Ribosomes will help to read this information, and complementary attachment will be happening of the nucleotide in order to bring different amino acids and attach them to each other. And this is our growing peptide chain, which later on will be attached to different 3D shapes, and this is what will be protein later on.